All right, in terms of the receptors, sensory receptors that are in the skin, you have tactile mechanoreceptors, or what they're called. You have Merkel discs. And those are actually up in the epithelial layer, or let's call it the epidermal layer. Meissner corpuscles, Ruffini endings, Pacinian corpuscles, and Krauss end bulbs are generally all down in the dermal layer. And so Meissner Merkel discs, they respond to touch, so the tactile receptors. Meissner corpuscles also respond to touch and vibrations as well. Ruffini endings, they are like skin stretching, deformation within joints, warmth. They're present in what's called both glabrous skin and hairy skin. Glabrous means skin that doesn't have any hair on it, say like your lips. The Pacinian corpuscles, those react to vibration. And the Krauss end bulbs, they detect cold. So I'm not going to ask you all of what each of these things do, but you should know about Meissner corpuscles and Pacinian corpuscles because we see those a lot. And I'm going to show you actually here in a second some actual ones here in some tissue sections. Next slide. I just want you to know that these receptors are there, and that's what's sending the sensory perception from your skin to your brain. So we talked about the nervous system, and so how did that information get there? And this is how it happens. So here's a corpuscle of touch, Meissner's corpuscle, and the lamellated or Pacinian corpuscle down here in the dermal hypodermal junction layer, and the Meissner corpuscle sticking up here in the dermal papilla there. Okay, next slide. So here's the different types. You have Merkel's disc. It's right up here. These are actually modified epidermal epithelial cells up here. They're right on that edge. Meissner corpuscle sticks up here in the dermal papilla. You have the Krauss end bulb here, Ruffini endings, and a Pacinian corpuscle down deeper in the dermis. And it's really close, often can be found right at the hypodermal-dermal junction. Next slide. Here's another slide just showing these. So we have Merkel cell fibers showing the cells there, and then we have the Meissner corpuscle, Ruffini endings, and lamellated corpuscles. And you also have, point out this, you also have free nerve endings up here. Those are often thought to be associated with like pain sensation. And then you notice all the axons of all the sensory neurons weaving through there. Okay, next slide. So here's just a cartoon of the whole thing. So here's your epidermis, dermis, and hypodermis showing the cold and heat sensory receptor, Meissner corpuscle, nociceptor sensing pain, and Pacinian corpuscle, they are sensing pressure or vibration. Next slide. Here's actually a Meissner corpuscle right here. That's what that is right there. So it's sticking up in that dermal papilla, and then there'll be a nerve fiber coming out of it, going back up to the central nervous system. Next slide. And this is a picture I took out of some slides we have in lab, and so right there is a Meissner corpuscle. I'll show a magnification of that, a couple magnified views here in the next slides. So you can see it sticking up into that dermal papilla region. Next slide. There it is again. And here it is again. So even closer. So there's your Meissner corpuscle sitting right there up in the dermal papilla. So this is slides we have in lab, and unfortunately I would love to show you guys these things, but we can't. And you'll notice down here, you notice all the dense, irregular connective tissue, right? So this is all dense, irregular connective tissue. And then we have all your epidermal layers up here. So we have your stratum basal. I'll just draw it here. That'd be basal. This would be spinosum. This would be granulosum. This would be lucidum. And this would be corneum here. And you notice that the lucidum layer, you see it looks sort of translucent. all through there. It looks like it's glowing in a way. So that's why it's called lucidum. It's sort of like making a glowing layer. Okay, next slide. These are what Pacinian corpuscles look like. They look like, you cut an onion in half. So they're all layers. And so what are the layers? Well, in the middle of this, right there, that is an axon. And these layers all around it are all connective tissue. So you have what are called fibroblasts, the cells that make connective tissue. They make fibers, and they make these layers all around an axon. The axon is unmyelinated. Okay, remember, myelin is layers and Schwann cell wrapped around, making layers around the axon, which would be about, the Schwann cell wrapped around that would be about like that big there, or about this big. Okay, so it's unmyelinated, which means it still has a Schwann cell there, but it's not like wrapped around it in multiple layers. It's like around it once. Okay, it kind of wraps the Schwann cell reaches out and it kind of wraps around the axon like that. Okay, so it's not wrapped around it like we showed before. So these are unmyelinated, but it does have a Schwann cell around it, but it ends up in this structure here, and this connective tissue, when it shakes back and forth, it will allow you to detect vibration, high frequency vibration, and pressure. So you can see how if it compressed, it could send a signal into that axon. Next slide. So here's an actual picture, more pictures of Pacinian corpuscles. So here's one here. You can see this right here. So this is your corneal layer. This is corneum, stratum corneum. And you see all your dermal papilla here, there, and there, and there, and there, and there, all sticking up there, right? So that means you have stratum basal, stratum spinosum, stratum granulosum, and it's hard to see a stratum lucidum there. So there's a Pacinian corpuscle there. You see dermal papilla are actually labeled there, arrow pointing to one. Okay, next slide. So here you can see the Pacinian corpuscle, this whole guy right here. This is all connective tissue. So there's connective tissue out here all the way around it. Next slide. And so here are the fibroblasts around it, and then the axon. You can see right there in the middle of it. So, and you can see all these little guys. Here's the nuclei of all, oops, how far with that? Just drawing a few of them. All the nuclei of all the fibroblasts that are making all these layers, all the onion layers all the way around the axon. Okay. Next slide. And you notice that this is clear there's a clear zone right around it. So that would be where the Schwann cell is wrapped around, mostly around that axon. Next slide. Okay. Here's another picture of the Pacinian corpuscle. And you got the axon right here in the middle. There's your axon. All these connective tissue layers and all these little dots. There. Those are all nuclei fibroblasts, if you can see all those. They produce all these fibrous layers all the way around this axon. Next slide. Okay, so we finished the skin, and now we're going to talk about the nail. So why are we talking about the nail? Well, it's actually part of the skin. So you guys talked about, I'm pretty sure you should have talked about, back in lecture about about proteins, talk about alpha helix and beta pleated sheets. An alpha helix is a structure that goes, so it's a protein structure that goes like that. Okay, so it's a very small thing, but you put enough of those together, and that makes, so you can see how that kind of structure could make a hair fiber. It's long and thin, right? Beta pleated sheet structure is more like this, and you can stick them together, and so you can have multiple sheets that fit together, and you can create that, and that could make a long flat structure. And so alpha helix, that'll make hair, and a beta pleated sheet, oops, beta pleated sheet that can make fingernails. So structure and function, they're almost always related. So these are both keratin structures, but you're making this with this beta pleated sheet kind of structure. So there's various parts of the nail. There's lots of names for everything. So we're in anatomy, so everything's got a name. So the free edge, that's number one here. There's your free edge. That's the part that's hanging off the end of your finger there. Then we have the nail body. Then we have what's called the lunula. That's the moon-shaped the white part down there near the cuticle, which is also called the eponychium. So that's the base down there where you push back your cuticles. And then let's see. So on this side we have the number 5. That's the nail matrix down here, and then we have the nail root. So you can see that's where the nail is actually being made, and then it gets extruded up onto the surface, and so it's exposed. And we're back to the eponychium, the cuticle right there, and then the lunula, then the nail body, and then the free edge. And then underneath we have what's called the hyponychium, also called sometimes called the quick. So if you cut your nails too far, you can cut them down to where the nail actually touches this underlying epidermal layer, and that's so you cut your nails back to the quick. You don't want to do that because it's kind of painful. The other things that are shown on the slide here, we have the epidermis, we have the dermal layer down below it, and then we have fat here, fatty layer, and this is bone. So that's the distal phalanx. Okay, it's a finger bone, so it's distal phalanx. This is the last one out there. And that'd be a sagittal section through the finger showing all the pieces. So the hyponychium is the part that's underneath the free edge, and the eponychium is back here down next to the lunula. Okay? All right, next slide. Fingerprint patterns. You have, I'm going to go over this real fast. It's just arch loops and whorls. And so you can go look up fingerprint patterns. Those are made, depend on those dermal papilla sticking up there, and they'll group together, and you'll make various patterns. And the idea is that everyone's fingerprints are unique, and it's probably true. There's probably no one that has exactly the same fingerprints as someone else, because it's somewhat genetically determined, but also but mostly random is the way these things produce these ridges. But you can see these ridges provide various surfaces, especially if the oil on them, that you can grip things more easily if they're wet or slippery. But also since you have oil on your fingers, you also leave fingerprints, and so you can tell what the fingerprints are by the different patterns. Okay, next slide. There's this was from the old book. There's different amounts of hair and different structures that are found in different parts of skin, so you can identify skin in different locations. So the axillary region, you have hair, and you have sebaceous glands, but you also have the eccrine glands here, but you also have these apocrine glands down here. So, and let's see, I can't see. It always goes in back behind the sebaceous gland here, so that's where it deposits its secretions. So there's a sebaceous gland is there. And if you have hair, you'll have sebaceous gland, you'll have a hair follicle and all that kind of stuff. So this is axillary region, and you have this apocrine gland. That's going to secrete little apocrine secretions, little vesicles come off, and little pieces of the cell come off, and they go up the surface, and they have protein and a little bit of liquid with them, and the bacteria break them down and make you smell. So the bacteria will create a smelly substance that gives you body odor. So the sole of your foot, you notice that has very thick corneal layer, thick corneum. You don't have any hair, so no hair. So if you look at two cross sections of things, you can see that axillary region. If you found apocrine gland and a little bit of hair, and some hair, versus something that had a thick corneal layer and no hair, you say, okay, those are two different regions. And then here's from the forearm, and you see a lot of hairs here, and you also see eccrine glands, sweat glands, and you have these sebaceous glands going around the hair follicle there. But what you don't see, so there's no apocrine glands there. So there's no smelly glands there. All right, next slide. So we have a lab experiment that we would normally do, and so which is a lot of fun to do, I think. And you can actually do this at home if you have any iodine or betadine solution. You can pick some up at Walgreens. What you do is you paint a little bit, paint you a little rectangle on your arm, and let that dry. And while you're letting that dry, just don't touch yourself, but touch your clothes with it. Just let it dry, and then mix up a little bit of cornstarch with a little bit of oil. It doesn't have to be a lot. And you take a Q-tip and you paint that after that betadine is dry, paint that over it. And what you want to do also is, before you start doing this, is go out and run around a little bit, and so that you get a little bit sweaty. And so what happens is you put some betadine on there, and you put some starch on there. Starch is in oil, and this starch and iodine together will react, and iodine will stain the starch a blue, black, very dark color. And it may take 10 minutes, 15 minutes for this to develop, and if you leave it on for half an hour, it'll develop even better. The starch and iodine won't really react until you sweat, so until there's water there. So the iodine and starch reaction, water is required for that. So that's why you use oil, so it won't react until there's sweat. So you have all these little sweat pores will produce a little bit of water, and comes out on the surface, and so everywhere you have a sweat pore, you'll get a little black dot. And so we're going to see what that looks like. Next slide. So here's my arm. I did this in lab. This was actually last year. And this is so here's the position of all the eccrine gland, or a lot of eccrine glands, on my forearm, on the inside of my forearm. So which would be the in anatomical position would be the anterior part of this would be my left arm here. And if you look on the back behind here, we have these models in lab. This is a large plastic model, and it's showing this white thing here, there, and there, and here. This is an eccrine gland, okay? And so there's the pore at the surface of the skin. So it's spitting out a little bit of the sweat, which is water with some salts. And so wherever there's water, the iodine and the starch react, and you get this little black dot. So, and that's what it looks like. Okay, next slide. So here's a close-up view of the eccrine gland pores on my skin. So you can see all those little tiny black dots on there. And that's the location of all the eccrine glands on the surface of the skin of my forearm in that little area. So pretty cool, huh? All right, next slide.